cartilage and blood, yes?
Yeah.
And we've got muscle and nervous tissue. So this is a section of skeletal muscle, and there are three kinds of muscle for you guys to be able to identify, be able to describe anatomically. So the first thing you notice here is this is a tissue that needs describing. And so if I want to describe the anatomy of a tissue and anatomy and physiology, I know that I'm going to have to pay attention to the arrangement of protein. So this is skeletal muscle, cross-section of skeletal muscle, and you can see that it has a kind of striated or striped appearance to it. The reason for that is the arrangement of two proteins that we will talk a lot about this semester. So this is your first introduction to them. Those two proteins are actin and myosin. A-C-T-I-N, M-Y-O-S-I-N, actin and myosin. So it is the arrangement of these two proteins that describes the physical appearance of the skeletal muscle. Those are the proteins also that I will use to describe for you the actual function of the muscle. How does it in fact shorten? And we'll talk about the interaction between those two proteins in order to do that. Now, there are some other things here used to describe these, and I don't think, frankly, that this is the best way to do it. Your textbook says that the cells are multinucleate. Now, that's a fair observation that you need to know. But when identifying these things under a microscope slide, it is not very easy to identify the borders of each individual cell, because oftentimes the cells are longer than what you would have when you look under the microscope. So it's true, they have more than one nucleus per cell. That is typical. But in order to identify it in the laboratory, that is not the best way. The striated muscle, you identify it because it's striated. Now, here's a second huge way of thinking and thinking about describing muscle tissue. This tissue is cardiac, and it is described exactly the same way as striated muscle. In fact, when we start talking about muscle stuff later this semester, I'm going to bring this up again, because the arrangement of actin and myosin is the same, which means functionally the way a skeletal muscle cell works and the way a cardiac muscle cell works is the same. But when you looked at this picture immediately after I showed you this picture, you didn't think to yourself that's the same, right? This does not look like that. The only way you could say that they're identical to one another is if you could take a cropping device and you crop this picture right here, and you look at that square, and then you crop this picture right here and look at that square. Now they look the same. The arrangement of actin and myosin are the same. But when you back off of it a little bit, you can clearly see that there are differences. What is the first thing that jumps out at you when you look at this picture compared to the other one?
That was messy. That was messier.
Messier. Yeah. So it looks busy. Yeah? What else?
A lot more white.
A lot more white in the picture, okay? Now, if we're going to use that messy and a lot more white in our description, we might say it more sciencey, right? We might say there appear to be gaps between the cells in the cardiac muscle, whereas the skeletal muscle cells appear to be stacked. And that is a very important observation between the two. When you look at a skeletal muscle, you discover that the muscle is in fact connected normally to a bone on this side and a bone on this side. And these fibers run between them, and when they shorten, they pull those two bones together. The origin, the insertion moves toward the origin. That's normally the way the skeletal muscle works. When a cardiac muscle contracts, what is it pulling on? You have to have something to generate enough force, right? You've got to have something for the cell to be hanging on to pull on. But the heart itself is not attached to any bones. So what we know is this. that there is dense fibrous connective tissue laced between the cardiac muscle cells, creating a fibrous skeleton within the heart mass itself. Places where the cells can hang on to something and pull on and push on whenever they contract. That's how the heart generates its force, because it is interlaced with a fibrous skeleton. And when you look at it histologically, it jumps out at you. That is the heart skeleton right there. Those are the bones, if you will, that the heart is contracting against. It's what they're pulling on, the connective tissues between the cells. So that's an important sign. When you're looking at these, you look at cardiac, you compare it to skeletal, one thing you see is there's space here. It's messy, and that space is filled with fibrous connective tissue. So you can tell when you look at them. Now here's another very important observation that will help you identify the heart over against skeletal. All through this tissue, you can see dark purple lines. Those dark purple lines are lines that are connecting cardiac muscle cells to each other. Now, this young lady asked me before class, she showed me a slide, and on the slide it had tight junctions.
And that's what I asked you, what they were.
What is that tight junction, right? That was the question, okay? So, in my description of the tight junctions before, see if you remember some of this, I said, anywhere in the human body where you have something hollow, the inside of it, the lining of it is going to be what? Epithelial. The middle of it will be okay, that's on the outside. You're one ahead of me. So if I go into the respiratory system, if I go into the digestive system, if I go into the blood vessels, is this all sounding familiar? The middle of it will have smooth muscle there. And I said, now, as we finish this, I'm going to show it to you. So that's the next picture. It's coming up, smooth muscle. And I'm going to say it again. The outside of it then is what? Connective tissue? Yeah, so this is prototype in anatomy. If it's hollow, it's lined with epithelium, smooth muscle in the middle, connective tissue on the outside. Like what? All the respiratory tubes, digestive system from top to bottom, all the blood vessels, all the lymphatic vessels, the uterus, the kidney, all the urogenital tubes. You expect to find this anatomy. All right? Now, in the blood vessels, as you get to the tissues where you are going to deliver what the tissues need, that is, the plasma and the nutrient inside the blood vessel has to get out of the blood vessel into the tissues to supply it with what they need. So as we get to the tissues, we find that the smooth muscle in the connective tissue is lost, and you end up with just simple squamous epithelium. Now, does that sound familiar? So in the lungs, right? One layer of simple squamous is the alveolus. One layer of simple squamous is the blood vessel. The distance the gases have to travel is very small, very efficient that way. Now, this is coming to the answer to your question. In some places in your body, specifically like your brain, you don't want to allow just anything to cross the blood vessels. So the capillaries in your brain pay very special attention to what can get across and what cannot. And they are joined by what are called tight junctions. It's like each cell is fused to its neighbor, like a zipper. And the pieces and parts of the zipper, the little pieces and parts, are proteins. They zip the cells together, forming a tight junction. So when you say the blood-brain barrier, you're talking about a tight junction. Now, the next word on that slide says desmosomes. And I'm going to talk about those later today when we talk about the layers of skin. And then the final one you should be aware of, which is why I brought it up here, is this one. This is another specialized kind of junction between cells. In this case, the junction is between cardiac muscle cells. These are called gap junctions, or intercalated discs. They are also proteins, but they are proteins that are fused into the membranes of each of the neighboring cells, creating a hole between them. So it's like cell 1 and cell 2 are right next to each other. They're not connected by tight junctions. They're connected by a protein, several of them, that create a hole that makes an opening between the two cells. So whatever you do to cell 1, you're going to do to cell 2 because they're connected. Now here's why that's so important. In the heart, let me start with skeletal muscle. In skeletal muscle, whenever you do something with a muscle, you want to use enough cells to do the job. So if I pick up something very light, I want to use a small number of the cells. If I pick up something very heavy, I want to recruit more and use more of the cells. So skeletal muscles have the ability to use some and not others. But we don't want our heart to work like that. We want our heart to work as a single unit. Every time the heart contracts, it needs to be at its peak efficiency. One contraction, all the blood comes out into the aorta and the pulmonary trunk. In clinical terms, those are called ejection fractions. And they're measurable. So your heart is supposed to squeeze out 70 milliliters of blood out of the left ventricle every time it contracts. The question in physiology then is, how does the heart muscle work as a single unit? You see it now? All of the cells are connected to each other. So when you stimulate the heart muscle mass, you stimulate all of the cells, and they contract as a single unit. It's called a ventricular syncytium. The whole mass contracts and forces blood out. This is why it can do that, because of these specialized junctions. And in the histology section, you can see them. If you look at a heart thing, the first thing that jumps out at you, it's messy, gaps in the middle, it's got a dense fibrous skeleton in it. The second thing that jumps out at you is these dark-looking structures that are connecting the cells, the intercalated discs. Cardiac muscle has special junctions. So have you heard of them all now? Tight junctions you can describe. Desmosomes are coming in the skin. Gap junctions are here in the cardiac muscle cells. Specialized junctions between cells. All right? Here's the third type of muscle. This is the smooth muscle cell. Smooth muscle cells are found in the walls of hollow organs mostly. They're found in the walls of hollow organs. In fact, I think we should extend that. We should say smooth muscle is found in the walls of hollow structures. Okay, so if we call the bladder an organ, then okay. If we call the uterus an organ, then okay. They're hollow, and we find smooth muscle in their walls. But as it turns out in human anatomy, smooth muscle is found in the walls of all hollow structures. It is, in fact, the regulation tool for the control of the size of the openings. So let's just do a brief little tour around this thing and talk about this. Smooth muscle in the walls of things. Can you tell me, class, what is the major reinforcing tissue in the airway, in the trachea? What is the major tissue that keeps the airway open? Okay, I didn't hear it. What is it? No. Who said that? What is it?
Hyaline.
Hyaline cartilage. Remember, there are 20 C-shaped hyaline cartilage rings that keep your trachea open. It's like a reinforced garden hose. Y'all remember that? Okay. Now, as you proceed through this hollow place, the cartilage disappears, and it's replaced with smooth muscle. Now, asthma patients know about this because anything that makes that smooth muscle contract will narrow the airway. So the drugs that are designed to help people with asthma or with some kind of acute allergy are drugs designed to make the smooth muscle in the walls relax in the airway so you can breathe. Now, if you swallow something and it goes down your esophagus and gets into your stomach and passes into the small intestine, how does whatever food, stuff, chyme, product that's there get from the stomach to the anus? I mean, we could have done it in the esophagus as well, but most people think, well, if it's in my esophagus, gravity will move it down. Gravity is not the only player there. The wall of the tube is moving, right? You guys are fans of Miss Frizzle and her Magic School Bus? Are y'all too young for that? No? Y'all know what I'm talking about? You remember the episode where the young boy eats the Cheeto and she shrinks the bus down and they go in on the Cheeto and she travels down through the digestive system pointing out to them structures there? I use this illustration all the time because it's such a great learning tool for kids. Miss Frizzle's Magic School Bus. So what she doesn't point out in that is that the bus would have been crushed at some point passing alongside the Cheeto because the walls of the hollow tubes have smooth muscle in them. And they contract circular and longitudinal, in alternating fashion. A process called peristalsis that moves product through the digestive system. This is the tissue that's doing that, the movement. Last one, and we'll complete our survey here. Drugs that are intended to impact blood pressure go after this in one of two primary ways. They either go after the control of fluid volume or they go after the control of the lumen size. Fluid volume and lumen size. Those are the two major ways to go after it. So if you want to do a hormone thing, like an ACE inhibitor, then you go after the process that keeps fluid in you. Some kind of fluid pill or something to make you get rid of excess liquid, and that will help with the pressure. Another way to go after the blood pressure is to go after the size of the lumens of the blood vessels, because some people have inherently smaller blood vessel lumens. And if I take 5 liters of liquid and I put it inside a hollow place, and I cause the inside of that place where the liquid is, a closed system, to be smaller, what will happen to the pressure? If I have a balloon, if you will, it's filled with water, and I press down on that balloon, what happens to the pressure in the system? It goes up, right? I increase the pressure in there. This is the exact same phenomenon that's happening inside of you with your blood vessels. So there are drugs like calcium channel blockers, major blood pressure drugs, that are designed to make the smooth muscle in the walls of your blood vessels relax. If the lumens are larger, the pressure is lower. Y'all see that? So this is another major way of impacting blood pressure. And it comes from the knowledge that we have about the walls of the vessels having smooth muscle in them. All right, last one is nervous tissue. Nervous tissue is made up principally of 2 kinds of cells, neurons and glial cells. So this one labels a neuron for you, and then it labels the nuclei of a supporting cell, and we will refer to those as glial, G-L-I-A-L, or glia, plural, G-L-I-A. The word means glue. But I'm going to teach you a bunch of them. The last test section of the semester, We will talk about a lot of nervous tissue. The third and the fourth, we'll talk a lot about nervous tissue and the cells there. For today, the main functional cell type of nervous tissue is the one that identifies it. You can see the neurons there. And those neurons have an anatomy that is typical. They generally have processes attached to the cell body that bring information to the cell body. And we call those processes dendrites. And they have a singular process that carries information away from the cell body. And we call those axons. So the neurons have dendrites bringing information to the cell bodies. They have axons bringing information away from the cell bodies. These are the communicating cells of the nervous system. We're going to spend a lot of time on them this semester, talking about how that happens. The glial cells are support cells, and there's a bunch of varieties, and we'll spend about 10 minutes during a particular lecture that I have designed for you talking about what glial cells do. They are not the cells that are communicating. You say, my brain tells my muscles to do something. The glial cells are not doing that. So the conductive cells are the neurons. All right. So we have left today and Wednesday and maybe a little bit of time on Friday to finish skin chapter, and then we'll be ready. Your textbook, I'm going to follow pretty close your textbook here, but I have listed a series of topics here that will show up on a couple slides in the presentation that I don't want to forget to talk about. Of course, the most important thing to start a description of skin is to talk about its anatomy. Okay, so that's the first thing I'm going to do, describe the basic anatomy of skin. In my description of the skin anatomy, I try to find some things that are interesting to talk about. One of them that your book does not mention is GSR. This is lie detection, so I'm going to bring this up. We'll also need to talk about skin color. I'm going to spend a little time here talking about melanin and hemoglobin and keratin, things that impact skin color. And then the rest of this will come sort of clean up at the end. Things about hair loss and acne with sebaceous glands, 3 kinds of cancer that are most common, basal cell, squamous cell, melanomas, and then burns. I like for my kids to finish their description of skin by being able to talk about the rule of 9s. How much of the body has to be covered with a burn before it's considered life-threatening? And how would you estimate that in a burn victim? Something used clinically today called the rule of 9s. So I'm going to teach you that as we finish this. Okay, so the anatomy first. Your book mentions some golly gee wow numbers about what you would find in a section of skin. And I don't ask my students to memorize these numbers. It's not important. Suffice it to say this. There's a lot more going on here than you might think. There's a lot of anatomy. There's a lot of anatomy in a square centimeter of skin. A lot of stuff going on here. So let's just talk general terms here, and then we'll start to detail out the specifics. The first observations I want to make are observations that should be very familiar to you. From outside in, young people, from outside in. The outside we call epidermis. What kind of tissue is that? Stratified squamous epithelium. The next layer in is dermis. What kind of tissue is that?
Dense irregular.
Dense irregular connective tissue. The next layer in is hypodermis, or superficial fascia. And that is primarily adipose. And then what will we find is associated with that superficial fascia?
Loose.
A deep fascia, which would be
Loosely irregular.
loose areolar connective tissue. Right. So these are things that you've already heard. All right. Now, I'm going to pick them apart now. I'd like for you to know a little bit more about the epidermis. And then we're going to talk about structures that are found in the dermis. The sensory parts, the erector pili, the different kinds of glands, eccrine, apocrine, sebaceous glands, and detail these things out. Okay, so the epidermis first. The epidermis in thick skin has 5 layers. You should learn them in order. corneum, lucidum, granulosum, spinosum, and basale. There they are. 1, 2, 3, 4, 5. So let me make a couple of comments about each of them for you. The stratum corneum is oftentimes described as a dead layer of keratin-filled cells. Now, this is also reviewed. When we looked at stratified squamous before, I said there are two varieties, right?